Hello, everyone. My name is Smita Gopinath. I am an assistant professor in the Department of Immunology and Infectious Disease in the Harvard Chan School of Public Health. And my lab studies the vaginal microbiome and its effect on infectious diseases and host immunity. And I'm here to tell you a little bit today about that subject and really try and convince you that there are some really exciting basic biological questions that we don't fully understand about this incredibly unique community, and that investigating those questions has the potential for immense impact on public health. Okay. So to begin with, what do we mean by the microbiome? So our mucosal surfaces, which you can think of roughly as any part of our body that is open to or interacts with the environment. And since our body plan is very much a tube within a tube, one of the largest surface areas is really the intestinal tract. And all of these mucosal surfaces are occupied by a complex ecosystem of bacteria, viruses, and fungi. And we call this together the microbiome. But really, our tools for studying these are probably the most advanced in the case of bacteria. So a lot of sort of microbiome can often refer to the bacterial community. And for the purposes of this talk, that's actually what I'll be referring to. But I just want you to keep in mind that that's just one component of this ecosystem. And as we get better tools, I will be able to investigate all of these interactions as well. And so when you look at that, each of these mucosal sites really have their own microbiome. And probably the site that we know the most about is the intestinal tract. And we know there's this really large, complex community of bacterial species that's incredibly diverse. And this diversity is strongly linked to human health. Additionally, this community really varies between people. It's influenced by diet, it's influenced by where you live. And what you can probably say is that your microbiome is more similar on different days between yourself and your household members, but it's hard to draw conclusions beyond that. The vaginal microbiome, however, looks quite different. And part of that is, let's say, what does the microbiome look like? So over here on the left, we have a wonderful picture taken by Carolina Trucchini from BBC. And what you see here is a picture of what the colon of a mouse looks like. And what you see in green is your mucus, and red and orange are your bacterial species, and in blue are your eukaryotic cells. And what you see right away is this really nice separation between your bacterial community and the eukaryotic self. And we know a lot about what happens when that separation goes away or how these bacteria might interact with your cells. But when we think about what this looks like in the vaginal mucosa, we really have very little idea. So on the right, you have really a smear from vaginal lavage. And what you've seen here is a large pink epithelial cell that is shed by the vaginal epithelia, as it's a stratified squamous epithelia. So you have your epithelial cells sort of being extruded and shed. And it's totally sort of covered with these rod-shaped bacteria that are lactobacilli. And we don't really have an idea of what the spatial structure of this looks like or what this looks like inside people, but we do know a little bit about what the bacteria look like. And in fact, we've known this for a while. So what do we know about the vaginal microbiome? This was actually, these rod-shaped lactobacilli were first described in 1899 as daughter-limb bacillus. It wasn't really until the 1930s that they were identified through metabolic activity tests as Lactobacillus acidophilus. And more specific species were identified with the onset of sequencing and PCR in the 1980s. And what's interesting is that really you see these lactobacilli in people across the world. So it does seem to be quite common and consistent, in sharp contrast with what we see in the gut. And the other interesting aspect is that this is hormonally driven. So on the table below is actually from that 1928 paper by Nat Stanley Thomas, where they took vaginal lavage from children and found that you did not see lactobacilli in the majority of those smears. And many, many studies since then have shown that your lactobacilli dominance in the vagina is really associated with onset of menarche. So this is puberty-driven and hormonal-driven bacterial community that, again, is very consistent around the world. It wasn't really until the late 2000s that people use the tools of 16S sequencing to look at vaginal bacterial communities. And what they found was sort of incredibly impressive and really fascinating from a biological perspective. So this is sort of Jacques Ravel's groundbreaking study that first identified what vaginal bacterial communities might look like in women of reproductive age in the US. And I'll just orient you to this graph a little bit. So this is with pretty standard 16S sequencing. So what you're seeing here is that in every vertical row, you have a single individual, while in the horizontal rows across, you're seeing different species of bacteria. So what you'll see right away if you're used to seeing these, from the intestinal tract, is this is a much smaller community. You really have not more than 30 species identified. And the colors in the graph show you relative abundance. So as you reach red, you're reaching close to 100% abundance, while yellow is closer to 1. And so you can actually start to make out patterns in here. So for example, let's take the one that has the most diversity and the most color. So you see over here, there's a community of people that have very few lactobacilli. So you don't see a lot of colors up in the higher top part of the chart. What you do see is a lot of sort of pink and dictating like relative abundance of a bunch of bacterial species that are highlighted by the black bar. And this is probably the most diversity that you would see, these kind of communities where you don't have a lot of lactobacilli, but you do have a lot of these other species, and they're incredibly high diversity. And in really contrast to the rules of ecological systems, the high diversity in this case is actually associated with poorer indicators of health. And we'll go a little into what that might mean. And then you sort of, when you work your way through the other groups over here, what you start to see is groups of people whose vaginal microbiome is really characterized by dominance of a single species. So over here you have the red bar indicating that people within this group have a highly dominant presence of Lactobacillus jansenii. Likewise, in this group, Lactobacillus crispatus. Over here, Lactobacillus inners. And then finally, Lactobacillus gasseri. So even within Lactobacillus dominance, you really have a number of different Lactobacillus species that are dominant within that community. And this paper sort of called these community state types. And this is something that has since really in the last decade been confirmed across the world. So there have been sequencing studies in multiple different countries identifying some subset of these communities. So you might not find all lactobacilli species, but you do tend to find broadly either lactobacilli dominant low diversity communities or high diversity non-lactobacilli communities. And it's pretty consistent in reproductive age groups. So when you think about this as a bacterial community, it's really quite independent of diet and really human adapted. The other aspect that's quite unique is that it is very, you don't find these in other animals. So our primates and our near ape cousins do not have this consistency of lactobacilli dominance in their vaginal bacteria, which really begs the question of how, what are we gaining from this? Having this community be adapted to human health, what exactly are they, how are they affecting host immunity, and what can we understand about the biological mechanisms around this? So of course, when we think about hormonal-driven adaptation, you can start to think about pregnancy. And people have seen that lactobacilli are strongly correlated to better pregnancy outcomes. So in this paper by Fetwide that came out in 2019, what they did is they sequenced the communities of people who then went on to give birth either preterm or defined as less than 36 weeks or at term. And what you see here is that while you do see some lactobacilli dominance in the community that gave birth preterm, it's much more enriched in people who gave birth at term. And they actually did metagenomics in this community, which led to this fascinating graph where they were actually able to look at what genes associated with individual bacterial species. So what you're seeing here is towards the left is term birth, towards the right is preterm birth, and each of these dots refer to genes within these bacterial species, and the darker the dots are, the more significant they are. So while they did see that some of the species in that more diverse cluster we talked about earlier, Prevotella, Smita, were associated with preterm birth, what really fascinated me is that the largest cluster associated with term birth, so giving carrying your pregnancy to term was Lactobacillus crispatus. Again, really suggesting an incredibly strong correlation between these species of bacteria in the vaginal tract to pregnancy outcomes, which are really sort of spatially separate and happening in the uterus. And these sort of differences, the other important part to know is that the Lactobacillus jenseni, gasseri, and iners clusters do not as strongly correlate with birth at term. And this sort of differentiation between individual species of lactobacillus actually carries through to other clinical correlates. And as an example, a set of studies coming out of a cohort in South Africa saw this was conducted by Doug Kwan's lab, so very similar communities. So in this case, they call them cervicotypes. So you have cervicotype 1 associated with crispatus, 2 associated with dominance of inners, and 3 and 4 were sort of 2 different groups that had these high diversity non-lactobacillus dominant communities. And what they were able to do with this cohort is actually, for a period of years, track incidence of HIV. And the results were really quite stark. So what they saw was that in the group of people with crispatus dominant microbiomes, they saw not a single incidence of HIV over 3 years. This was not true of people with inners or people with the high diversity communities, really suggesting that the specific species dominance in the vaginal tract is strongly correlated with resistance to, in this case, HIV, but there have been other studies extending this to other sexually transmitted infections. And sort of as an example, the other correlate in here is when you look at vaginal inflammation, again, having crispatus dominant microbial communities in the vagina is correlated with less pro-inflammatory cytokines present in that vagina. So if we were to sort of take a pause and put together everything I've talked to so far, you could think about vaginal microbiomes being divided into 2 categories. You have your lactobacilli dominant low diversity categories, and you have your lactobacilli low high diversity communities. And in this lactobacilli low high diversity communities, in contrast to what we know about the intestine, this increase in diversity is actually strongly correlated with negative health outcomes, specifically increased acquisition of sexually transmitted viral infections. There have been studies also showing increased transmission because you look at virion load within the vaginal tract. You see increased rate of preterm birth, as well as increased basal vaginal inflammation. And there's a lot of sort of groups that really study the associations between specific bacterial species to try and understand how they drive vaginal inflammation and potentially increase acquisition of STDs. On the other hand, when you look at these lactobacilli dominant low diversity communities, you really see that the flavor of lactobacillus species that you have is also pretty important. While broadly speaking, again, in this case, in stark contrast to other microbial ecosystems, low diversity is strongly correlated with health and resilience, but also that the specific species you have is quite important. So you see over here that Lactobacillus crispatus in particular is strongly associated with healthier outcomes. And so what happens to the sort of balance between these 2 groups of communities? And you can start to ask yourself, if you have one community, do you always have that? How resilient is it, and how long does it stay on within a particular person? And to look into that, I have to introduce you to one of the most common really medical syndromes that afflicts people, which is called bacterial vaginosis. And it's really sort of a fascinating immune mystery, and it has really high clinical consequences. So on the right, what I'm showing you again is a vaginal smear, and you might recognize this from the slide earlier that I showed you. So on the healthy subjects, what you see are these epithelial cells surrounded by what are gram-positive rods or lactobacilli. But when you take a smear from someone with bacterial vaginosis, what you see instead are gram-negative cocci that are really overgrowing and present in the smear. And one of the fascinating things is this balance between gram-positive lactobacilli and gram-negative high-diversity communities is actually a clinical criteria for diagnosing BV. This is called the Nugent criteria. BV is also incredibly diverse. It can look very different in different people. And in fact, if you take healthy subjects, you would find absence of lactobacilli. The Ravel paper earlier only looked at healthy people. So you can actually see non-lactobacilli high diversity communities present in healthy people, but it is also highly correlated to poor health outcomes. So understanding that shift between the presence of these bacteria and lack of symptoms, and understanding what sort of drives then symptomatic bacterial vaginosis is something that is really an open question in the field. As I said before, BV is associated with loss of lactobacilli dominance and increased anaerobic bacteria. And it really remains unclear if it's sexually transmitted, because there are multiple people who see an increase in incidence of BV after sex, but also people who don't describe that association. So it's sort of very much an open question. So why do we care so much about BV, right? So it's really, in addition to the symptoms being really painful and lowering quality of life, it's a risk multiplier. So a single BV episode can increase risk of multiple STDs, including chlamydia, gonorrhea. It can independently increase your risk for HIV acquisition along with having herpes. And it's really thought that there's something that BV does to the vaginal immune homeostasis, because even if you, say, treat having herpes, another sexually transmitted infection, which increases your risk for HIV, and even upon treatment of herpes with acyclovir, you still have increased HIV acquisition in people with BV. So there's something sort of fundamental about the vaginal immune homeostasis that's changed in BV. And the really depressing thing about this is that BV is still treated and the same way for the last 70 years, which is with antibiotics, and the recurrence rate can be up to 60% in particular populations. So this is something that not only is debilitating for quality of life, but actively increases your risk for multiple outcomes. I didn't mention here, having BV also increases your risk for preterm birth. And we really don't have good solutions to help that population of people who just get recurrent BV. And I would posit that understanding more about the vaginal immune homeostasis would really help us look at what is driving the harm associated with BV. So given all of this, a reasonable question to ask might be, well, what can we do to manipulate the vaginal microbiome, given that we know what is associated with health? And I can tell you a little bit about what's been done in this area as well. So in 2019, there was a randomized trial of Lactin-V, which was basically a vaginal suppository of specific Lactobacillus crispus communities, and they gave a pretty high dose, listed over here, for over 10 weeks. And the results were positive, but with a very small effect. And so what people saw, in fact, was that while it did reduce recurrence of BV by 15%, if you looked further out for another 10 weeks, in less than this particular strain was only found in less than 50% of people, suggesting that community change is even in a simple and small community is really quite difficult. And I want to also pause and say here that oral-based community change, so basically taking probiotics orally to change your vaginal community, really has been shown to not work. This trial was done with a vaginal suppository, which at least introduces the bacterial community directly to the mucosa. But as we can see from this trial, we really do need to understand more about what it takes to affect change. There has been a single study of a non-randomized vaginal microbiome transplant, in which they took vaginal fluid from people and transplanted them to people with recurrent BV. This was done with really just 5 people. And what you can see from here in terms of abundance, the darker blue says Bifidobacterium, and actually Gardnerella, which is a bacteria strongly associated with bacterial vaginosis and lactobacillus. So what you can see here is with A and B people, a single dose was sufficient to increase lactobacillus dominance, at least out to about 4 months. But with 3 out of the 5 people, they really needed repeated dosage. And for one of them, even multiple dosages did not seem to work. So we really need to understand more about what drives community change and what is the heterogeneity of factors between different people and their microbiomes. There is the first randomized vaginal transplant study ongoing right now in Boston area. It's called the MOTIF study, and it's run by Drs. Carolyn Mitchell and Dr. Doug Kwan and Dr. Fatima Aisha. And I strongly recommend going to their website and checking it out if you're in the area, because they are looking for both donors as well as trial participants. And I think this study will really tell us more about what are the factors that, is it possible to transplant a whole community when you also add environmental factors. So unlike a probiotic suppository, this would be taking mucins and whatever other factors and sugars might be in the lavage, and hopefully help that community to flourish. And I have really high hope that we might learn something from this, and I encourage you to go check out the MOTIF study. All right. And so when we think about what it takes for a vaginal microbiome transplant to work, we can ask, what does stability look like in the vaginal microbiome? And we have a great way to look at that, which is the occurral menstrual periods. So for example, here, this is a paper that sampled women during menstruation for about 4 months. The red dots at the bottom of the graphs refer to a menstrual period. And what you see here is abundance of bacteria. And what you see, this is a Crispatus dominant subject. And right around these red dots, you see a drop in Crispatus and an increase in this case, Streptococcus. However, at the end of the menstrual period, Crispatus comes right back. And this is something that we're really trying to mimic when we think about community change. And you can see these in other subjects as well. So for example, here you had somebody whose community did not change at all during menstrual period, but remained Crispatus dominant. However, when they did undergo a temporary change between menstrual periods, again, Crispatus was able to come back up. Menstrual periods can also be a time of community shifting. So in this case, this subject had an iners dominant community that bounced back after several menstrual periods, but after the last one indicated over here, underwent a community change to that more diverse lactobacilli lost community. And in fact, this paper found that inners in general tended to be a gateway community out of lactobacilli dominance into loss of lactobacilli in general. So I would really posit that we need more of these kinds of menstrual study clinical profiling to be able to understand how lactobacilli communities bounce back from this natural occurrence and perturbation. And if we understand how these bacteria are doing that, we can take advantage of that to better capture community change, to be able to push people towards healthier microbiomes. And with the last really 10 minutes, I want to talk a little bit about what we know about vaginal antiviral immunity within the vaginal mucosa. And so when you think about the vaginal mucosa, really dominated by these bacteria, also fungi and viruses, but you understand less about this, and you think about how the microbiome protects you from infection. So if you're a mucosal pathogen, you will encounter the microbiome before you encounter the eukaryotic host. And these interactions can really influence infectivity and transmission. And for the most bacterial pathogens and the bacterial components of the microbiome, these bacterial interactions are between two alike organisms. So broadly, a microbiome can be protective against invading bacterial pathogens. However, we might imagine that this looks quite different for viral pathogens. And one of the things that my lab is really quite interested in is understanding the interactions that drive this microbe communication at the mucosa. But there's also the cells at the mucosal surface, and being a stratified squamous epithelia, the vaginal mucosa also has these small numbers of immune cells that circulate and sort of traffic in and out of the mucosa. And one of the things we found pretty early on was that you don't need a lot of immune cells to be activated to generate protection of the mucosa. This is work that was published. I'm not going to go too much into it, but we actually found that giving a bacterial small molecule that was a toll-like receptor ligand that activated less than 1% of these specialized adaptive immune cells, in this case, dendritic cells in the vaginal mucosa, and activated a host of antiviral genes transcribed within these cells. And that 1% of cells activating these genes was sufficient to protect the entire vaginal mucosa against both genital herpes simplex as well as Zika virus. And so this really speaks to looking beyond interactions with the vaginal epithelial cells, but actually studying how vaginal microbes might be impacting these really small immune populations within the mucosa to better get a sense of how they alter immunity. And in the last 5 minutes, I'm going to tell you one small story about a vaccine technique that we hope will really sort of lead the way to understand more about inducing and activating immune cells within the mucosa. And to do that, I'm going to highlight one particular antiviral gene in here that's also a chemokine. In this case, this is the chemokine CXCL10, which we know from a lot of previous work can recruit T cells into the vaginal mucosa, where they can then become something called tissue resident T cells, where they stay within the mucosa, don't circulate in and out, and provide really lifelong immunity. And one of the reasons to target these kinds of cells is that your mucosal immunity is really quite different from your circulating immunity. So when we think about how we test for vaccine results, you really look for factors of vaccines working, whether that's antibodies or activated T cells in circulation. And that may or may not be a good reflection of mucosal protection. And in fact, we know that in the case of herpes, there are multiple vaccines that induced really high levels of circulating antibodies and activated T cells that still completely fail to protect. And so in light of this, Akiko Iwasaki and Hannah Shin first came up with the idea of prime and pull as a vaccine technique. And the idea was to take vaccines that had succeeded at generating lots and lots of circulating antibodies and activated T cells in circulation, but failed at mucosal protection. And by understanding the chemokine cues, you could then rescue them by pulling all of these cells that were activated and in circulation to the right mucosa. And that's why they call this sort of prime and pull. And what's great is that in the last couple of years, we've shown that at least for the vaginal mucosa, this works really well. So you can, they show that it was protective prophylactically against genital herpes in mice by generating local memory T cells. And since then, there have been 2 studies coming out showing that you can also protect therapeutically. So actually prevent or reduce herpes virion replication in individuals that already have herpes. And as an example of this, we use the guinea pig to study this. So this is a image of the guinea pig mucosa, and the green dots here are reflective of virus-specific CD8 T cells. And what I want you to see here is here's your vaccinated guinea pig that has very few T cells, but just application of a pull compound, you can increase T cell density in the vaginal mucosa where they need to protect against invading viruses. And you can also see these guinea pigs develop herpetic ulcers, very similar to what we see in people. And you really see that here you're comparing between the green vaccine-only arm and the purple vaccine and pull arm. You saw far fewer lesions and herpetic ulcers in guinea pigs that got the prime and pull vaccine. And what we were able to show is that we could use this really cheap small molecule, which actually is also an antibiotic. So we found that aminoglycoside antibiotics activate dendritic cells in the vaginal mucosa to really induce antiviral immunity. And just application of this neomycin antibiotic protected mice prophylactically against acute infection equivalent to chemokine. And one of the reasons this is exciting is really that chemokine or protein really can be expensive and labile and require a cold chain, but this is really off-the-shelf small molecule that would be pretty easy to apply topically as a pull agent. That really extends this technique into this vaccine technique into something that can be carried out pretty easily. But how does this then relate back to the microbiome? So I would actually posit that while it's important to look at the members of these communities in here that are driving inflammation, equally that there are these lactobacilli-dominant populations that are associated with increased antiviral immunity. And you could start to think about what other small molecules are being secreted by these communities over here that might affect host immunity in ways that we really just haven't found out yet. And so if you want to put this together on the concept of how the vaginal microbiome might be affecting antiviral immunity, on this side, you have a very On the right side of this picture, you have a very diverse, high diversity, low lactobacilli vaginal microbiome that induces lots of inflammatory cytokines. And we still don't understand how these inflammatory cytokines drive increase of susceptibility to infections. It's a little bit easier with HIV where you could say, oh, they actually end up calling more T cells, and that serves as a host for more virus. But that doesn't quite explain the increased susceptibility to other STDs, including chlamydia, gonorrhea, and herpes. Conversely, we see with these low diversity vaginal microbiomes, we might imagine that they are actually secreting things that actively alter the immune environment within the vaginal mucosa, and that investigating these communities could really serve as a potent way to mine for novel immunomodulators. And that, in fact, in the last minute is something that my lab is working pretty heavily on. So we've been trying to identify ways that the microbiome influences the host immune response, both by mining these lactobacilli for novel compounds, and also by trying to really make artificial sort of mouse models that are colonized with human lactobacilli. As I told you, lactobacilli are unique to people to sort of begin to understand how they affect host immunity. Most of our best immunological tools are really built in mice. So we need to find a way to get these bacteria to stay within the mouse vagina so we can understand how they influence host immune responses. So that's something that we're actively working on within the lab right now. And with that, I'd like to take a moment and thank members of my lab. I wasn't able to talk about their work today, but we have several exciting projects on these interactions, and we think we've identified a novel immunomodulator that I hope to tell you more about later. And with that, I hope you enjoyed the talk.